Okay, so I think I want to start, and I don't care whether we are 2 or 0, At the end, I want to deliver my class, and I have to say I have a lot of fun preparing this class because it's one of the topics that we are going to discuss today. To me, are very kind of inspiring, and they belong to the core of what is now synthetic biology. So before, I hope that the ones that are not here today can still see the record, because I insist that today I think is an important lecture. Okay, so before I start with the topic today, I want to share with you some of my coordinates, because obviously I don't want this to be just a situation in which I just come here, lay an egg and run away, right? So I really want to maintain a degree of contact, and these are all my coordinates. So you have, I'm sitting in the Systems Biology program of the National Center of Biotechnology in Madrid, my email and everything, my webpage where you can see many of the things that we do. We even have a lab video that was recorded time ago, so I appear a bit younger, but part of that, the topics that we describe in this lab video are the same. I'm also very active in Twitter, and if you happen to be in Twitter, I don't ask you to follow me, but if you follow me, you will have interesting information, because I use Twitter for communication of the results from my laboratory, for commenting papers, for sharing ideas and this type of things. Okay. Then, my laboratory, as is the case of other laboratories in my center, is all the time looking for newcomers. And newcomers involves undergrads, grad, and postdocs. And if you have interest to, at some point, considering my laboratory as a possible destination for either of these 3 situations, then please email me, and then we'll see what we can do. I know that in India you have a system through which undergrads are expected to spend some time, right, in others' laboratories. That does not work exactly like that in Europe. Okay? We have a different system, and our undergrads normally go to nearby laboratories. Okay, don't go somewhere else as you do normally. And also, having an undergrad in the laboratory involves some expenses, and they have to be covered. So sometimes it's not easy to bring undergrads to the laboratory, but it's not impossible. And then we have also students that come to the lab to make PhDs, and now my laboratory, what is very good for, is for postdocs. And because we have resources, and postdocs normally comes with their own ideas and so on. So at the end, is a good place for developing one's own interests and so on. Okay, so let's get deep into this topic that, as I said, I consider to be one of the most, I will not say interesting, but one of the core, concepts and activities in synthetic biology, around which a larger community has been formed in the last few years, in the last decade or a little more, and is this whole thing about circuits and how to develop with genetic tools, with genetic parts, devices and systems that develop some type of logic that is comparable to computing. I would not say it's rigorously, absolutely computing, because there are some issues that I would like to discuss later. But in principle, the idea is to see to what degree we can implement in bacteria or in other biological systems the ability to compute signals and respond to the signals with a deterministic response that is ruled by a set of logic rules or logic considerations. So the start of the field was this paper that I have discussed in the very first lecture, and it's about making E. coli to be like a kind of photographic film. So this hello world is one of the typical, sentences in computing when something new appears, hello world. And the idea in this specific paper that is now nearly 20 years ago was to see whether, by the same token that computer scientists develop new programs, new interfaces, new computing capabilities, we can use bacteria and biological systems in general, biological parts, biological devices for making something similar, with the hope that at the end we may be able to use, to program entirely biological systems as we can program our computers. Okay, so the basis for this idea of programming and making circuits with bacteria, with biological systems, is identical to the ideas of using electronic devices and electronic circuits and all the rest of it. So, well, as you come from engineering to some extent, I mean, this is not new for you, but you know that in electric engineering, you have like 3 critical parameters: voltage, current, and resistance. And you can make a metaphor on how one is compared to the other. For instance, you have here water, and you have here a valve that closes or opens the valve here, the current. Then you may open this, but then that you open this, that not means that immediately everything passes. You have to go through this narrow neck that is equivalent to what we call resistance in electric circuits. So this is how things look like in an electric circuit. So you have a voltage, and you have a ground here, and then the logic of the system is that if you don't have a connection here, then there will not be a kind of current coming in this way. But if this switch is closed here, then you have an out current that goes. So this switch can be implemented mechanically, like with a hand or something, or it can be implemented with a transistor, and of course transistors that, as you know, are the basis of advanced electronic circuits and the rest of it. So this is something that you may find in books, you know, what is resistance, what is voltage, and what is current, each of them with the corresponding parameters. And one open issue that appeared at the beginning of synthetic biology was whether there were formal equivalents for amperes, for volts, and for ohms. So you can speculate that at least one of the parameters, that this current could be associated or could be made equivalent to this concept of POPs, polymerase per second, that goes from one promoter to the other. But reality, as you may have figured out before, is that in biological systems we don't have these electronic devices. So the idea is how to implement materially the same logic that we can implement in electronic circuits, but using as our tools, our little components, exclusively material parts that come from biology. And the most important of these parts, there are others I will tell you later, but the most important are transcriptional, say, regulatory assets that, as we have repeatedly addressed in previous talks, for the most part go as promoters that are either activated or are either inducible upon a signal. So in some cases, a promoter will be repressed by a signal, by a repressor, and you have a situation of negative regulation. In other cases, you have just the contrary. You have the situation where the promoter by default is off, but then you, through transcriptional factors and inducers, can turn it on. Okay. So with these simple Lego parts, so the idea is to construct complexity, logic, and computation. So how can we do that? Okay, well, I will mention on passing some things that we have mentioned before, but now we will go into more depth, okay? So the super basic unit of every circuit is the so-called ANOT logic gate, okay? And just to refresh your memory, this is the way you can implement a logic gate with a electronic, say, situation. You have a resistance, you have a transistor here, and then you have two inputs. You have one input, and then the input that goes out, the signal that goes out is inverse in sign in respect to what gets in. Okay, so the logic is 0, 1, 1, 0. We have discussed that many times. How you do that? Well, there are many ways of doing it. The most typical thing is to play with repressors, and to play with a repressor that represses another repressor. So repressor to repressor means positive signal. Okay. So there are numerous cases that different synthetic biologists have made these constructs. And for instance, in this case, that one was very well characterized. What we had is a PLAC promoter that is repressed by LAC Y, but the promoter is de-repressed when IPTG is added. And then this promoter controls the expression of the lambda repressor that then in turn represses another promoter. That is the target for the lambda repressor. And you have a ribosomal binding site in front of VAP. So you have a super typical inverter. When you have as a signal IPTG, then you lift the repression here, and then the repressor, the lambda repressor is produced, it represses the second repressor, and then you have an inverse function. Okay, so this is repressor means activation. Okay, you can then go on and go into more complex gates, and one that we have in fact studied with some detail in this course is this proposition of a AND gate that involves two inputs, and you need the two inputs to have an output. So in this case, if you remember, one of the inputs was a promoter that expressed a variant of the T7 RNA polymerase of phase T7 that had two stop codons that could be overcome upon different expression from a different promoter of a suppressor tRNA that was engineered under the control of a promoter responsive to salicylate. Okay, so in such a way that only when you have a defective polymerase and something that suppresses the defect of the polymerase, you have an output here. Okay, AND gate, typical. There are many that you can make. Then you can make, you can compose different gates. For instance, you have here an N AND gate, where you have another type of AND gate. In this case, it's based in a promoter that depends on a factor called sigma 54, but in any case, it's a promoter that can be activated by either one transcriptional factor or the other, and then each of the transcriptional factors have been engineered in front of a promoter that responds respectively to either IPTG or to arabinose, and then you have an AND gate. But then the POPs that comes from here goes into a NOT gate, similar to the one we had before. So at the end, the logic of the system is an AND gate, meaning by that that you have positive outputs, whether you have known one of them, but you don't have any output, you have both outputs here. Okay. You have then go on, for instance, making these NOR gates. NOR gates are very important, as I will tell you later, because they are the basis of many more complex circuits, and they can be implemented in many different ways. So in this case, for instance, the way to implement a NOR gate in which the only way to have an output is when you don't have any input 0, is, for instance, where you have a promoter here that can be repressed under a repressor, but then the repressor can be in turn be expressed through two alternative promoters, one or the other. So whether we have one or the other, we have repressor, and then the repressor will repress this promoter here, and therefore we know output. But if none of these promoters 0 are there, then there's a chance, and then the promoter, this downstream promoter will work. Okay, and you have a formal equivalence with this electric technology.
Are these circuits faster as electronic circuits?
No, they are much slower. I mean, everything goes in biology. Everything goes slower. I mean, you make a sugar, and it goes instantly. In biology, you put your system. You go to have a coffee or to have dinner, and then return and wait for some little change. Okay, so it's not so. Well, I'm exaggerating a little bit, right? But things are much slower in biology, right? Unfortunately. Okay, so on this basis of these simple and basic logic gates, what type of devices we can make that are similar to the devices that electric engineers use for their constructs? Well, I'll show you just some of them. Some of them we have seen, but I will go back to them. One of these is this famous or infamous repressilator. We have been through it. I will not get into that. But if you remember, the idea is that you have three repressors that repress each of them the next one, repression, and then as the dynamics of the system has some given parameters, then the situation is that at the end you have a cyclic behavior of the whole system. I will have dealt with that already, so I will not get into that. But there are other cases where you can also use transcriptional regulators, in particular repressors and also activators, to generate some oscillatory, pattern. For instance, if you have a repressor that self-represses its own promoter. For instance, imagine that you have like I, that is a repressor, and I is expressed through the very same promoter that it expresses, right? So also, depending on the parameters, you can have a situation in which the system will go necessarily up and down, up and down, up and down, up and down, okay? So because in moment number 1, the promoter might be repressed, but then the protein has a given lifetime, and then at some point it will go out, and therefore it will disappear, and therefore the promoter will be active, and then once it's active, it will repress the activity of the reporter gene that is somewhere else, but at the same time it will repress itself in such a way that over time you have this oscillation, these oscillations. Obviously, as we are in a biological system, the oscillations are not so perfect and not so beautiful as you have as an electronic signal, but still you have this kind of pattern and behavior that corresponds to this specific configuration. Well, you can go on, and instead of having an oscillator with one gene, you can have oscillations with two genes. For instance, in this case, you have here A and B. In one case is RSC, in the other case is LAG-Y. In this case, is an activator, in the other is a repressor, and then they are connected genetically, and we can make this connection through genetic engineering in such a way that the repressor self-represses and the activator self-activates. But then, at the same time, the repressor represses the expression of the activator, and the activator stimulates expression of the repressor. Okay, so well, you know, you have to think a little and to really see how the logic behind that is, but certainly this is one of the configurations that you can implement by making these logic gates in such a way that you have these dynamic interactions between a repressor and an activator. And indeed, the prediction that you get will be something like this, and what you have in reality is something like that. Okay, so it's not too bad, right? And you have these oscillations at the end, and well, this is something that again is not perfect, but at a distance could be equivalent to what electrical engineers would prepare with electronic components, following the same logic and the same types of interactions between something that activates and something that represses. Other type of device, the toggle switch. Again, we have discussed this already, and we will not get back into that. And remember that one of the interesting things of the toggle switch is that it enters into the system a kind of memory. Memory means that you are on and nothing happens, you will remain on and on and on and on until you come with the other inducer, and then the other inducer will turn the switch into the other direction. Okay? So again, we have seen this before. I will not get into the details because we have discussed them in great detail. But there are other types of circuits that are super interesting that are not based on transcriptional regulators. And they are based in a different principle that still are based on genetic parts or in DNA parts, biological parts, but the mechanism of performance is quite different. So let me introduce you in 2 minutes about this phenomenon that appears in all scales of life that is called site-specific recombination. So during this course, we have heard the word recombination in different contexts. So the most trivial context is homologous recombination. Homologous recombination is the phenomenon when you have a swap in the DNA strands in locations that are homologous to each other. Okay? So, and this is mediated by some very complicated mechanism called the REC-A mediated mechanism. But the thing is that of homologous recombination, the basis of that is that you need long stretches of DNA which are homologous to bring about recombination. There's this other type of recombination where you need much shorter segments that is the basis of another technology that we have seen in previous lectures that is called recombinering. So in recombinering, you just need a much shorter sequences to bring about the recombination. But then there's still another more precise way of recombination that has to do with very short sequences, and very short means 10, 20 base pairs that are enough to bring about recombination between homologous sequences that are present in a donor and in a recipient. And this happens because of the action of some enzymes that are called site-specific recombinases that are called integrases. So integrases can go in one direction or the other. So you have a segment of DNA, and then you have another DNA circle, for instance, that have these homologous sequences, these very short, by themselves, this will be insufficient to just recombine as such. But thanks to these enzymes, this circle can now integrate in the system, because of the homology of these two with these two. And at the end, you have the whole circle integrated there. So what happens, and this is part of the funny thing, is that when you have the two sequences going in this direction, what happens is that the site-specific recombinase may bring about not the integration or the deletion, or the deletion in that direction, but the inversion of the sequence that are between here and here. So you may start with the situation, and these are the sequences that are recognized by the site-specific recombinase, and then because of the action of this recombinase, then you may end up with a segment that is identical to what you had before, but turn around in terms of their relative orientation. Okay? So these site-specific recombination enzymes are good for flipping DNA in one direction or the other. And then some variants of these site-specific recombinases can go in both directions, and in other cases they can go just in one direction. So smart people have thought that, well, this ability to kind of flap around and to switch around a sequence of DNA can be used to generate logic gates as well. And for instance, there is this interesting paper that I'm going to comment briefly on how to implement a short gate by making, through not transcriptional regulators, but by making use of these site-specific recombinases. Just to refresh your memory, short gates are those in which you have an output only when one of the two inputs are there. You have no inputs, nothing happens. You have the two inputs, nothing happens. But when you have only one input, then something happens, right? So let's see how these people thought that you could bring about this logic with site-specific recombinases. So what they did was the following. You have a promoter here, and then downstream of the promoter, you have a reporter gene that will help you to monitor what is going on. And then what you do is that between the promoter and the reporter gene that is here, you put a terminator. Okay? A terminator terminates transcription, and therefore, if the terminator is active, the transcription cannot proceed into the reporter gene. But then what you do is that you flank this terminator. Well, there's another aspect of the terminator I have to say, is that terminators in many cases work only in one direction. If you turn around the terminator will not work and will allow transcription to proceed. So only you have the terminator in this orientation, the terminator will work as an actual transcriptional terminator. Now, you flank this terminator by two sequences that are recognized by one integrase. There are many integrases, many types of site-specific recombinases. But in this particular case, the blue ones are recognized by integrase A. And flanking these sequences, you have other sequences that are being recognized by other recombinases, another integrase, okay? And therefore, at the end, depending on the integrase that you express in the cells, the integrase will act on these sequences here or will act in the other sequences, okay? So now, the starting point, that means no signal, okay? If you have signal, you have no integration, no expression of the integrases, and therefore the terminator will stop termination because it's in the right orientation. But what happens when we express the integrase A? Okay, we express it in trash. Okay, what happens is that now it will flip around and the terminator will go into an orientation that will not be active. And therefore, what will happen is that transcription will proceed. Meaning by that is enough that you have just one input, one of the integrases, and you have here expressed, right? Okay. So what happens when what happens now? Okay, when you express the other, we'll return to the previous to the starting point. What happens when you express the other recombinase? Okay, well, exactly the same. So it will flip the sequence downstream of the promoter. Obviously, not between the blue triangles, but between the green triangles, and the terminator will be inactivated because the orientation has changed, and therefore what will happen is that transcription will proceed. What happens when we express the two terminators, the two recombinases? Well, what will happen is that one will compensate the other, and therefore now the transcription will stop. Okay? Yes.
Is it that one compensates the other, or is it that one rotates and then the other also rotates?
Exactly. So that it's a purely geometrical problem. It has to do with parameters. So the advantage of this case is that the system will work regardless of the parameters, because you need to have a promoter that is stronger or weaker. It's just that you are in one direction or the other. You cannot adopt any intermediate location. So it's very smart and very kind of interesting, right? Because you can have this complex logic by playing with a system that naturally can only adopt two orientations. So it's a super digital thing. Either you are like this or you are like this. You cannot be like this, right? And therefore, this was a quite kind of interesting thing. How to make this bigger and combine it with other gates might be more complicated, but in any case, things may develop differently. On this basis, synthetic biologists over the years have proposed different types of circuits in the immense majority of circumstances based on transcriptional factors, because are the ones that appear to be more handy. And I'll show you just a couple of them that I find quite kind of interesting. So, for instance, there is this article proposing a circuit that will be a push-on-off switch. What is a push-on-off switch? That it's like you press one, it's on, again off, again on, again off, again on, again off. So in this case, the input signal is UV light, and then UV light is known to be connected to various, readouts here, and to various, they affect the expression of different regulatory, factors here that were reshaped, and I don't think it's worth that we go through all the details here because it's too complicated. But basically, we have two promoters here. One of them goes in the direction of producing 3 repressors, and the other go into the direction of producing 1 additional repressor and one reporter gene. But then they are wired genetically through all these interconnections. So this one produces this one, and then it represses this one, and then another is produced here, and then it goes here, and then it goes here. So you have all this circuitry that by the time it was made had to be made by hand, say by just using some type of intuition. And then the result is this state in which, depending on the illumination, you illuminate once, and then the cells make this peak of activity. Then you illuminate twice, and then the activity goes down. You illuminate again, and it goes up again, and you illuminate again, and then it goes down again. So obviously, well, these guys reported only 4 cycles. Okay, so I suppose that you keep on pushing and pushing and pushing and pushing. At the end, the system will stop working. But I find again very interesting that by using a logic that is typical of electric circuitry, like pushing in and out, you get a situation in bacteria that resembles this kind of a scenario, right? So it's kind of cool. There are more things. For instance, there's this article a bit later or a bit before, I don't remember. There is a counter. A counter means that depending on the number of pulses that you show, then the system will give you a response that will indicate you how many times you have tried the system. In this case, you have a promoter that responds to a ribose switch. That is another regulatory element that I have not discussed in this course, but it's a regulatory asset. And then it will interact with a ribosome binding site that will enter the same polymerase. The polymerase will go to the same polymerase. We go to another RBS here and a GFP. And the idea is that if you add the signal once, then you get this protein. If you add it 2, you get this protein, right? So you have this circuit can count up to 2. In this case, things are more complicated, and then the circuit can count up to 3. So depending on the number of times that you add the inducer, then you have proteins of different colors. Okay, so that's something that, I mean, counting to 3 is not a super big deal. 1, 2, 3. I mean, any little baby can do that, right? But it's curious because bacteria normally don't do this type of operations, of mathematical operations, and it's kind of intriguing that you can program this in your place. But, and this is a big but, so far all these circuits that I told you were based on intuition, right? Okay, you sit around, you have a brainstorming, or you think on an IM project, and you think, well, how can I implement this counter, or how can I make this push on, push off, whatever. But then, this is not enough. You go to more complex circuitry, you cannot base that on intuition only. So on that basis, years ago, in fact now nearly 10 years ago, someone called Chris Voigt, and I think we have mentioned his name various times, developed this platform that has become super useful for the automated design of genetic circuits. Automated design means that you, in an abstract way, decide what type of logic you want to have in your circuit, and depending on the behavior that you want to have at the end, you have to program some connections between different logic gates, and so on and so on. But then, instead of now thinking and scratching your head and say, well, where can I find all these elements here to implement the circuit? Then the idea is to have a platform that will allow you to go directly from your original design or your desired design, instantly converted into a sequence of DNA that can be put in a bacterium and expect that it will work as expected. Okay? So the kind of dream situation, the kind of ambition is, as I said, to have a tool that is quite independent of the user, and the user does not need to be a super expert biologist, in which you just decide the logic that you need to program your bacteria for developing or manifesting a given phenotype, and then what kind of sequence at the end you have to produce to make that happen. Okay? Well, this one important consideration in this whole story about automatic implementation of genetic circuits, and is something that I mentioned on passing one of my previous talks, and it is that in any logic gate, even if it is very simple, you have to ensure that the inputs and the outputs happen in a range that can be understood by the next operation. So if you have a range of activity where you have, well, obviously every promoter has an on state and an off state, and then, but you don't go directly from 0 to all. Then you go through some kind of gray zone. And then the idea here is that you decide that you are in an on state when you are above a threshold, and you are in the off state when you are below a threshold. But then these thresholds, the up and down, can be now interpreted by the next step as positive or negative output, right? Because if you go beyond or below, then the connection will not work. And to do that, you need to generate a considerable number of data, in every promoter and every transcriptional factor and every regulator that you use in this type of constructs, and you have to know that data to automate the production of the different complex circuits. Okay, yes. Okay, so this is exactly what is represented here. So, for instance, imagine that you have this would be two inverters, one after the other. So imagine that the input signal goes from here to here, and then the next one understands the signal from here to here. Okay, so the differences are big enough, and the parameters are good enough so that you can make the connection and secure and predict that the logic will be a perfect inverter after inverter. But if it happens that the variation of the input goes between here and here, and the variation of the output has to go from here to here, it is evident that this variation will not be sufficient to enter a change in the next one. Okay, so that means that you may create a large collection of inverters, but then you have to qualify each of them and to secure that the different inverters, in terms of parameters associated to inputs and outputs, can talk to each other, because if not, they will not work. So what Chris Voigt made was an immense effort to make a large collection, like 20 many repressors, and then make all possible combinations, and then at the end decide what, making a chart indicating what tables or what gates were compatible, where were put upstream or downstream, and then during this compatibility study, then you have some combinations of inverters that are possible to put together and others that are not possible to put together because they will simply not work. Okay. So this is then the kind of flow that you get with the Cello workflow with the system. So first of all, you have to generate data on the behavior, on the parameter associated to the behavior of the promoters and the repressed promoters in vivo through cytometry, and you get parameters in off and so on. And then you get some data here that you can say curate and make a model to make it more or less compatible with a model. And then, well, you have that in a repository, in a database, and then the beauty of this system that so far has not been improved by any other concept is that the user has an interface where you specify what you want, okay? And then run, and then the system, and you will not see that, will look in the database of parameters associated as in every time of repressor and regulatory system that you have at hand or that has been studied, the degree of compatibility between the different ins and outs in such a way that it will process and will make different models of different possibilities, and at the end it will appear with a proposition of a sequence that in principle should deliver the same logic that you have programmed here. So in this case, the thing is that you don't have to think on what promoters I'm going to use, what regulators I'm going to use, because the system will just do that for you. Okay, so this is really fantastic because you see that how a complex circuit can then be implemented by the system in the shape of a DNA sequence that you can have it synthesized and then you test it in your system. The thing is that before the final construct is implemented, it may happen that you find different solutions for solving the same problem, because you have many possible promoters and many possible repressors and many possible combinations. So typically you say, well, please write the sequence of DNA that should be optimal for this particular circuit. And then the system goes through the various possibilities, and then it will give you a kind of a score of what possibility would be better for your specific purposes, depending on the data and all the combinations and all the comparative analysis of the behavior of the individual gates. So the same system will make simulations of what would be the best possible combination for your specific, say, construct, and then there are probably the system will return all possibilities that go beyond a certain score, and then it's up to you to decide which of them will be, in principle, more, will be better for that particular purpose. Obviously, the one that appears with a better score in connection to what you want to do at the beginning will be the first that you may want to try. But as we have discussed previously, sometimes the context may change a bit the parameters and so even though ideally this would be the solution to your problem, in some cases it's not enough to test just one possibility, but you have to test various of them and then select in vivo the ones that is closer to your interests. So let me see. Yeah. Okay, so I'm going to show you a super easy a case for construction of a NOR gate with the Cello system. And in this case, the Cello system has the virtue that is applicable not only to E. coli, but also to many other bacteria, provided that you have data for the different repressors that you have in your at hand for making this type of combinations between different regulatory parts. So in this case, we wanted to construct a NOR gate. So why we're interested in NOR gates? Well, NOR gates are the super basic unit of every single electronic circuit that we have all around us, you know. So our telephones, everything, for the most part, are mostly based on NOR gates. So, and the NOR gates, as any other gate, it should have an input, some outputs, and they have to be connected. They have to be connected so you can build complexity. So the first step was to go to a library of repressors and then determine the parameters through which this particular repressor is able to repress a target promoter here. So we had a bunch of repressors, and then all of them were under the control of a PTAC promoter that can be induced by IPTG. And then you have to generate data through which that will tell you how different repressors will repress with different intensities the cognate promoters that will in turn give you a signal that you can monitor as part of the experimental workflow. So when you have that, what you do is to examine the compatibility of the different gates with others. You know, what you have here is the list of repressors that we have tested, and what we have here is you know, the score and the compatibility of some of them with the other. So after in E. coli, you know, when you do this in E. coli, you have many compatibilities here. When you are in P. putida, you don't have many compatibilities, but you still have five scenarios that the system will tell you that you can implement with a good chance that the logic of the system will be that of a NOR gate. Okay. Okay. So then before you go ahead and synthesize the DNA, this is one of the best solutions that the system will tell you that if you use this particular repressor called SRPR, and this particular repressor and the other, wait a minute, and the other, there is RAC, there is a transcriptional factor, and you have as inputs IPTG and arabinose, then at the end, the type of logic that you will get will be this one. That is exactly the situation that you want to have, because you want a NOR gate. You know? So this is the prediction, this is the simulation that the system will kind of do on the basis of the data that you have already entered for each of the particular parts. And then this is what happens when you make the real experiment. So remember, you know, in the simulation, optimal conditions, you have only a significant output when you don't have any of the inputs. What happens in reality? Well, when you implement this segment of DNA, because you have it synthesized, you send it to a company to synthesize it and put it in one of the SEBA plasmids and put it in P. putida, then you have the logic, more or less, but you have to be careful with this context variability, because if you implement the same segment in a low copy number plasmid, then you get the logic, but well, the logic is not perfect. However, if you implement that in a medium copy number plasmid, then you see that the logic is much better. Okay, so that means that you can make a kind of pre-assessment of a good sequence for producing a good logic gate, but then at the end you have to keep an eye on specific parameters to ensure that at the end the thing will work more or less as you want. Okay, so, but you know, I think that this simulation, the UI perfection, is not very different of what we get under given conditions. So I think that the system is very good, and as I said, so far things have not been really improved a lot in respect to what happened before. But then you can really go further and further and further in complexity. And just to give you an example, the same laboratory published a few years later a system that converted E. coli in a digital display. Okay, so in this case, what you have is this typical, you know, scheme where you have A, B, C, whatever. You have you have 7, say, geometrical shapes, and if you remember, in every digital display, you can get any number depending on whether you fill all of them. If you fill all of them, you have an 8, but you have F and E, you have a 1. If you have F, B, and B, C, you have 2. I mean, you can do, or you have this 2 shape here. I mean, you can form any number based on that, right? So the electronics that is behind these shapes here is that the system works with 4 inputs, input 1, 2, 3, 4, or combinations, or 2 of the corresponding inputs, and then depending on the combination, the inputs that you have here, you may have a combination of outputs here that illuminates or gives light to each of the segments that you have there, okay? So this is pure electronics. Now, what you do? Well, you then try to implement with bacteria what type of, say, parts of this geometrical shape you have to fill. That means giving an output depending on 4 with inputs plus the combination of them, and what type of output you would get in every case. So what these people made with the cello system was to develop circuits for each of the outputs. Remember that we need 4 inputs, and in one case a combination of 2 inputs. And then you need to have, at the end, you have 7 possible outputs, and the shape and the illumination of different outputs will give you the shape of the corresponding numbers. Okay, so this is something that you can implement, you can model through in a completely simulated fashion, and see what logic gates you have to implement and so on, and then you ask the cello system to generate the sequences of DNA that will respond to that specific logic, okay, with all the connections and all the rest of it. And then at the end, this is what you have. Obviously, this is not perfect, let's face it, but it's not too bad, right? You can get, depending on whether you have inputs here, and there are 4 inputs, IPTG, 1 autoinducer, tetracycline, and another quorum sensing inducer. Then you have 1, 2, 3, 4, 5 is okay, well, a little weak, 6, 7, 8, 9, and 0. So in this case, you just combine, how different inputs will result in different outputs, and you put the outputs in a geometrical fashion so that the populations are separated, and then you get numbers, and that's something that, to me, this is I don't know if it is very useful, but conceptually, I find it fascinating, right? That you can really program E. coli to give you a number. I find it super, super nice. Okay, so but this is not the end of the story. Things can get more and more complicated and more fascinated as well. So far we have discussed only situations where everything happens in one cell, But in reality, we can do much better, because we can leverage the fact that in a population you have different types of bacteria, different types of cells, and then to implement what in a computation is called distributed computing, right? So it's like, instead of having a logic gate or a circuit being delivered, being implemented by just one bacterium, you take advantage of the fact that you have different types of bacteria to really execute logic functions and do things that are interesting. So in this case, for instance, well, this is what you have with computers. You can have everything happening in a single computer. You can have a grid where different computers are collaborating with each other to give a little computation, right? So in biological systems, one way out of the various, at least 2 ways to make bacteria to collaborate among themselves or to do something together for the sake of giving some logic and making some circuits and some computation, is through 2 schemes. One of them is through conjugation. So conjugation, again, this is something that we have discussed on passing, and is this phenomenon through which one bacterium is able to deliver a piece of DNA, in most cases a plasmid, to another bacterium here. What is the advantage of this situation for computation? That similarly to what happened with the site-specific recombinases, is an on-off state. I mean, either you get the plasmid or you don't get it. You cannot get it in between, right? So that means that you are in a 0-1, again, digital logic, because the plasmid is transferred or is not transferred. It cannot be half transferred. There's another type of tools for implementing this community-based logic, or this, say, distributed computation, and is based on quorum sensing. If you remember, in one of my first talks, we discussed quorum sensing as a mechanism through which bacteria note or notice that they are surrounded by other bacteria by means of secreting into the medium a compound that is called an autoinducer, that if you are by yourself, it will dilute out, but if you are surrounded by people like you, then it will reach a certain level, and that level will suffice to trigger a community response in a simultaneous fashion. Okay, so this is something that can also be leveraged for making circuits of various types. So going to what I said, so in the case of quorum sensing, then you have this autoinducer that goes out, and then the autoinducer will be recognized by a regulatory, say, protein that might be present in a recipient here, that is connected to a GFP, okay, so a reporter. So one of the strains release the signal, and the other, not necessarily the one that releases the one, has a regulator that will activate a GFP. But in the population, you have these two, okay? So you have one that is an emitter, and the other that is an emitter plus a reporter, okay? So that, as you will see in a moment, that is a tool also for making this type of combinations and logic gates and so on. But then conjugation is a lot better, because as I said, you have a plasmid, and you have a donor, and you have a receptor, and either you deliver the plasmid or you don't deliver the plasmid. There's nothing in between. Okay, so for different types of logic gates and circuits based on quorum sensing, there has been a number of papers, and in this case, for instance, is a situation that I also mentioned in my first talk on passing, in which we have a receptor, we have an emitter, and we have a receptor in the population, and then depending on the concentration of that emission from one of the cells, then the other population will sense that emission and will produce either a red protein in the case of the emitter, or a green protein in the case of the receiver. Okay, you can make these patterns that, well, are interesting. But also you can make other things. For instance, you can make, say, combinations between emitters and receivers, and then you put them in a plate, then you generate a kind of a boundary situation where, according to given parameters, you can produce specific shapes, okay? So if in this specific strain, where you have this quorum sensing based implemented response to a signal, you have like three inputs, like IPTG and TET, following a given logic. Then depending on what you put in every position, then you can have different letters being produced, right? It's like a, I don't know, this could be like a V, like an O as an X, J, H, U. So this would be reminiscent of what we had before with the other case of the numbers, but in this case, the implementation of the logic is not just one bacterium, it's a collection of bacterium. I mean, this is infinite, you know, the number of possibilities that you have for making, for using computation not only at the level of one cell, but also at the level of a population. I mean, it's just a question of imagination, everything that you can do. In this case, for instance, what you have is another situation, another circuit here, that in which bacteria, depending on their circumstances, oscillate. And to see that specifically, you have to put the bacteria in a receptacle to secure that the quorum sensing signal does not diffuse out completely. And then basically what you do by making this circuit is to have oscillations in the population here, in the intracellular, in the levels of release autoinducer that goes up and down. And then if you monitor fluorescence, for instance, of the cells that are in this chamber, they go like nothing, a bit, more, less, more. So you have a oscillatory behavior of the entire population, not just the behavior of one cell, as in the case of the repressilator or so, but in this case you have the entire population behaving in this oscillatory fashion. Also, I mean, you can engineer ways of having in a device, in a microfluidic device or other type of physical device, communication between cells that depend on emission into the media of quorum sensing signals that then will be integrated with other signals to get layers of synchronization. So this is something that, if you are interested, I mean, there are thousands of papers in this respect, but really I just want to show you this without getting into the details, just to show you the power of combining the logic of some circuits at the level of single cells together with the logic of systems when you put another ingredient, and the ingredient is this community-based, aspect, right? Well, I don't know, I have a bunch of slides here on this. I don't know how new, how much novelty it will bring to this discourse, but you know, you can play with two bacteria, you can play with 3 bacteria, you can really amplify the logic of what you want to do in a very, very dramatic way with more bacteria, and with circuits that are engineered in each of the bacteria that compose a population. Okay, so that's something that, for instance, in this case, what we have is a case where we have 3 cells, and each of the 3 cells will act as a NOR gate. because each of them have inputs and outputs that will be recognized by the others, in this case a pure quorum sensing based system, and then the emission of a signal of quorum sensing by one of the bacteria will be recognized as an input for the other bacteria, and then if at the same time you have another input that goes here and here, then you generate at the end this type of logic that responds to a short logic. But unlike the cases that I showed you before, where the short logic is implemented in one bacterium, in this case the logic is implemented in a community. Well, these are some details of this, how this could be implemented with different parameters and so. It may not be that important, but at the end the logic that you get is what I showed you. Also, you can make this edge detector where you you can produce a shape, and then the shape that you can generate, for instance, with this optogenetic system, and then you can make a bacterium that is able to highlight the positions of this shape that form an edge. I mean, the picture is not very clear, but you see that this is what you kind of illuminate in the plate, and this is the edge detector. So the system will detect only not the image itself, but the borders of the image, right? And then in that case, you have to construct another gate here, and I mean a little complex circuit with NOR gates and so on. And then you implement it, and at the end what you have, by playing with this population distribution of the computation, is exactly this type of detection that is kind of interesting as well. But you can go on and produce senders and receivers, senders and receivers, senders and receivers, and at the end you can end up with a population that can really compute as a whole population. In fact, we were, one of the discussions we had over coffee these days is whether bacteria could act as a kind of brain, And obviously, this type of manipulations does not mean that bacteria act as a brain, but the moment that you have a biological system that is able to compute signals and deliver responses with a kind of fixed logic, it starts to be reminiscent of a brain, right? So that's something that maybe in the future will have consequences. Well, the other possibility is to play with conjugation, as I said, and then you can also implement logic gates on the basis of different types of conjugation, and to have, for instance, you can implement this circuit here. And then the trick in this case is that you have a plasmid that will enter into the next train a signal that will be computed by the receiver, and then the combination of the two in turn will emit another signal that can be delivered to another recipient of the conjugation. And the basis of these interactions are not diffusion of an autoinducer, but are conjugative bridges. So I don't want to go into detail, and I think you have enough circuits, and it's a complex story. And but just to leave you with a take-home lesson that indeed you can use conjugation as an on-off switch that can be engineered for making some type of computation in vivo, right? Let me just say on passing, and these are just my last slides of this story, that logic gates can be implemented not only with transcriptional factors, not only with site-specific recombination, it can also be implemented with metabolic reaction. So in metabolic reactions, you know that one compound A might be transformed in a compound B, and that will be a yes gate if you want to make it abstraction. But in many cases, in a synthesis reaction, you have substrate A, substrate B, and they react and they form something else that is C. So if this C can enter as an input to another metabolic process, then at the end you can really use also metabolic reactions as a way of implementing logic. And this is something that has been used in various locations. A word of warning is connected to the fact that I have also mentioned to you that DNA is not always fixed. It mutates, and mostly when you have some stress, is frequent that the metabolic reaction to the stress will be production of ROS or production of some metabolic regime that will alter the performance of the DNA. Okay, so it's just a word of warning. Don't think that when you have a genetic circuit, it will last forever, it will perform forever as you program it, because it's likely that over time it will develop some mutation that will get you a bit wrong. Well, I had other things here, but I think you had enough for today. Yeah, so yeah. Okay, let me just, well, just on passing, you know, the thing is that I would have liked to have more time to tell you many more things about synthetic biology and the tools and everything. And some of these things I have to rush to them because we don't have the time. But I don't want to leave you without telling you that there are ways, for instance, to implement transcriptional factors that are, or repressors that are not natural, that are unnatural. Not only the DCAS variants that work together with the CRISPR system and that can originate a kind of synthetic repressors, but also there's another type of proteins that are called tail proteins that you can program to target specific locations of the genome, and therefore you can generate a whole bunch of different repressors that will target given sequences. So the number of repressors that you can use in circuits is virtually infinite, because you have many that occur naturally, but there are many others that are not naturally occurring but can be programmed themselves. And the tail proteins are good in the sense that you can, there's a rule through which you take a protein and then you can vary the sequence of the protein, and at the end the resulting protein will be able to repress transcription or to bind strongly to a DNA sequence, and therefore it will act as a factual repressor. Okay, so well, I have some other things here, but I think I will not get into detail. Other word of warning that I want to mention is that you know, at the level of single cells, we live in an stochastic world. Remember the discussion we had yesterday or a few days ago, in which we talked that the response of the bacteria or the response of the promoters to given signals may be, say, monomodal or might be multimodal, and then that introduces a level of unpredictability that is important to tackle. Okay, so I just want to finish this part on the circuits and so by directing you to this page here, Angel GM, and Angel is the director in my center of a laboratory that is computing with living biological systems. And if you get into this web page, you can have all types of information on everything that I've told you today, because he's been one of the pioneers in this type of approaches. Okay? So I leave you at this point. I think we should go for a coffee, for a break. We still have things to tackle. But yeah, so even though at the end, you know, I had to rush to these more complex circuits, I just want to leave you with the flavor that you can use a bunch of different biological parts and biological activities to build logic or to implement logic that so far were only possible with electronic circuits. And I think that that's something very good and that will have a bright future for the construction of biological systems for given purposes. Any question, any comment? No? Okay, so let's make a little break of 5 minutes and then we'll return to just round off this story about the circuits and so on. Okay? 5 minutes.